Swayam Prabha. Digital India. Educated India.
Welcome back to my course, Industrial Biotechnology. Now let me continue that. Now let me talk about the solid state fermentation process. The solid substrates are such as the wheat bran or pulp from sweet potato starch are used for the culture media, and then pH is 4 to 5. Inoculum in the form of spores of Aspergillus niger is spreaded as a layer. 3 to 4 centimeter thick layer formation is there. Then incubation at 28 degrees centigrade. Growth is accelerated by the addition of alpha amylase. Solid state fermentation time is about batch fermentation is 80 to 100 hours. The old liquid surface fermentation, because solid surface, liquid surface, both are surface fermentation process, but in liquid surface fermentation media will be liquid, and in solid surface fermentation the media will be solid. And here this is the oldest methods of citric acid production, simple technology, low energy cost, high reproducibility, and interference of the trace element and dissolved oxygen tension is minimum, and 20% citric acid in the world is produced by surface fermentation process by using liquid media. The nutrient supply is the meat molasses that is used. The fermentation carried out in the aluminum tray filled with sterile nutrient media. Then inoculum spores are sprayed, sterile layer was supplied, and then temperature is 30 degrees centigrade. Spore germination take place and layer of mycelia forms on the surface. The pH drop to 2 and mycelia grow in size and forms the thick layer on the surface of the nutrient. The fermentation stops after 7 to 15 days. The mycelia and nutrient solution was separated, and mycelia is mechanically pressed and thoroughly washed to obtain the maximum amount of citric acid. The nutrient solution is subject to processing for the recovery of citric acid. Final yield of citric acid is 0.5 to 0.7 per gram of sugar. This is the yield what we have in the liquid surface fermentation process. Now let me give you the more information on the submerged fermentation process, with largely in operation in the industry. And I work with Citric Biochemicals. We use the submerged fermentation process for the production of citric acid. Now 80% of the world's supply citric acid is produced by the submerged fermentation process. It is considered the highly efficient and easy automation. Disadvantage is the adverse influence of the trace metals and other impurities, and variation of oxygen tension, advanced control technology that required high trained personnel, because this is the kind of since it is the fast process, so you require little bit trained personnel for the operation of this process. Now two types of bioreactors are used. One is we have start tank reactor, another aerated tower reactor. What you call, I told you that the airlift fermenter is used, but it is less used in the industry for citric acid production. But bioreactors is made of high quality stainless steel, because the reason is that the citric acid has some corrosion property. So the quality of the stainless steel is usually used, the high quality stainless steel usually used. When I discussed about the material of construction, I told you that how the metal ion plays important role for improving the characteristics of stainless steel, particularly chromium and molybdenum, that increase the acid resistance and corrosive characteristics of the stainless steel. So this is the stainless steel 317 is usually used for the citric acid fermentation process. The adequate supply of oxygen, 20 to 25% saturation is required for the good production of citric acid. Now problem of foam formation, that is quite common, because since it is the aerobic fermentation process, the foam formation is quite common. So to have this to subside the foam, we shall have to use the anti-foam oil, and for the sterilized anti-foam oil we put it in the reactor to subside the foam. Now this is the flow diagram of how the citric acid fermentation process is usually carried out. This is the molasses from the road tanker, because it is come in a tanker, and from the tanker I told you that the molasses storage tank, what you call MST, that is located close to the main gate of the factory, and where this is a very big tank where we stored the molasses for throughout the whole year, because the molasses is a seasonal product, and since it is a good raw materials for the citric acid production, we need this. But we observed we have acute scarcity. The off season we have the acute scarcity of cane molasses, because in India cane molasses also used largely for the ethanol production. So we replace the cane molasses by using the hydrol. Hydrol is a byproduct of the glucose industry, that after crystallization of glucose, or that in the mother liquor contains about the 50% of sugar, that is replaced by the molasses. So we successfully replace a portion of the cane molasses during the off season, and we find suitable for the citric acid production. Then this molasses measuring tank, what you call MMT, that is located in the fermentation plant, and so whatever day-to-day requirement is there, we used to pump this to the fermentation plant, and this pump is the special type of pump I told you, because this is, since it is a very viscous liquid, we use the gear pump to drag the cane molasses from MST to MMT. And after that it contains about the 50% of sugar, so you have to dilute it, because in the fermentation media the citric acid concentration is about 10 to 11%. So we know that naturally we shall have to use sufficient amount of water for dilute it and maintain the sugar concentration. Then we shall have to add some kind of nutrient as per cell mass production is required, where we require sufficient nitrogen source and other material. So we put it here. And this we recycle again and again to ensure that this media is perfectly mixed, it is homogeneous. After ensure that this is perfectly homogeneous, then we pass through the medium sterilizer. In the medium sterilizer we take it here. So we have two vessels, we have inoculum fermenter, we have production fermenter. We can take it to the inoculum vessel, and we can take it to the production fermenter. Now here I want to again point out, I already mentioned in my previous lecture, mineral concentration in the fermentation media plays very important role. So cane molasses contains significant amount of iron and magnesium, so that is to be removed before we use this cane molasses for the production fermenter. For the inoculum vessel we don't require any kind of pre-treatment, but for the production fermenter we shall have to remove the iron and manganese. And here you can see the anti-foam oil tanker. This if we have sterilized anti-foam oil, this is we use for subside the foam. And this anti-foam tanker also connected with this mechanical seal. I told you when I explained the bioreactor, this mechanical seal that sapped also, and it is lubricated with the help of this anti-foam oil, so that friction between the shaft and the mechanical cell is minimized. Then after the so what happens in this fermentation process, that this inoculum vessel and production fermenter, they are located in the fermentation plant. So and usually the volume of the inoculum volume of the inoculum is usually 5 to 10% of the fermentation media. So, the Citric biochemicals where I work, the capacity of this reactor was 200 cubic meter. So, so naturally that this should be 10 to 20 liters. So, 10 to 20 cubic meter. So, our size of the media was around, we find the 14 cubic meter is good enough for the for this production fermenter. Now question comes that very interesting question, that how we prepared the seed culture in the lab for this fungal fermentation process, because we know that fungi can not only use for citric acid fermentation process but also use for in the different antibiotic fermentation process. Now all the fungi, most of the fungi, their filamentous growth, and you very difficult to you have to quantification. Quantification of the organism very important, plays very important role. In case of yeast cultivation, what we can do, we can count the yeast cells. So we can number this. We can what number through R&D, we research and development, we try to find out what number is will be most suitable for the production fermenter. So that number we can do easily in case of unicellular cell. Now question comes how we can do in case of fungal fermentation process where the cell has the filamentous growth. So what we do in this fungal fermentation process, we use to do the sporulation of the fungi. Because why sporulation of the fungi? Because spores you can count. We can under hemocytometer you can count the spore, and so sporulation of the fungi take place under stress condition. What is the stress condition we maintain? Mostly the moisture is the test condition. So I can show you the technique that is used in our industry, how we prepared the seed culture in the lab. So it is very simple. I can tell you that we can we have bread, that the kind of bread we have available in the market is like this, that is used during the breakfast. This kind of bread, a small bread we can have. So we can cut it with the help of knife. We can cut it. We can just cut it like this. So we can have 4 pieces. That small bread we can cut it like this. We can have 4 pieces, and then the white mouth this that test tube, that is cotton plug. This is cotton plug. Then here we put this. We put this bread, and then we inoculate the organism here. Now here, then the bread, you might be, I told you bread is produced by using yeast, because yeast is used, and yeast has vitamin B, which is required for the growth of required for the growth of Aspergillus niger. So when you keep it and inoculate this organism, so it has the limited moisture content. So after some time, this will be totally covered with spores. The totally covered with. Then we can make a suspension in 0.85% saline water, and then this we can find out that what particular concentration of spores is required for the inoculum vessel. We develop this particular concentration of spores, and then we put it in the seed can. I showed you the seed can, how it looks. The seed can looks like this. Now here we have a valve. Here we have a valve. Then here we have nut and bolt arrangement, where when you take it to the inoculum vessel, this nut and bolt arrangement we connect and we sterilize this line, and then aseptically we transfer the spores. This has the spores inside. Then and again we sterilize the line and open this nut and close this valve and open this nut and bolt and take it to the laboratory. This is how it can be done. Now another thing I want to show you, that I did my PhD from IIT Delhi, and during my stay there was an international conference, and there we demonstrated one experiment how to develop the spores by using different type of fungal culture. And what we use, the very interesting thing that I want to show you, this very simple technique through which we can develop the spores. Here we can take the barley grains. This is the barley grains we can take. This is barley grains. And here we put the honey peptone media. Honey peptone media. Then we inoculate the cell culture, Aspergillus niger, or anything, whatever fungal culture we inoculate this. Now we keep it in the incubator for some time. But one day, after some time, you will find mat formation. This is at the surface there will be mat formation. So what we can do, we can take the culture that bottle out, and we can strike the hand like this, because since it is a solid particle, as soon as we strike, then this particle will be this mat that is formed here, this mat that is formed here, that will be disturbed, and this cell will be coated on the surface of the barley grains. Then again we keep it in the incubator. After 6 hours again we take it out, again we strike it like this. After some times we find that each barley grains is totally covered with several spores. Then we make a solution, we prepare the 0.85% saline water and shake it and take the spores out. We can easily, any organism we can produce spore like this. So we can use particularly, you might be aware that penicillin is produced by Penicillium chrysogenum. That is also very good color of spores, green color. In case of Aspergillus niger, we produce the black color of spores. So beautiful, different fungi, they have different color of spores production is there. So this is after that. So you can see that since the capacity here at 5 to 10% as compared to production fermenter, we have different capacity air filter we use for the different purpose. Here should be 10 to 20 times higher, 15 times higher than that of the air that inoculum vessel. Then after the fermentation is over, you take it to the harvest tank. Now this whole unit remain in the fermentation plant. But after the fermentation is over, I told you how we take the fermentation broth to the harvesting tank. We just here we have a air outlet. In the air outlet, if you close, then there will be air pressure here. With this air pressure, even you don't have to use pump. Without using pump, you can drag the liquid from production fermenter to the harvesting tank. And when it comes to the harvesting tank, this is located in the recovery section or the purification section. Then it first passes through the rotary vacuum filter. You can see this is the rotary vacuum filter. When we pass it through this cell mass is to be separated. After during the fermentation process, cell mass is very important, because cell mass plays a major role for the production of citric acid. After the fermentation process, the cell mass is the impurities in the reaction mixture. So you have to remove the cell mass first by using rotary vacuum filter, that I shall explain in details. Then that filtrate, you take it out and put it in the filtered reservoir, and that you put a vacuum so that the air content here should be, oxidation doesn't take place. Then we further purification is taking place. I shall discuss you later. Now inoculum preparation, I was discussing that sporulation of Aspergillus niger is prepared in the laboratory under aseptic conditions. Spores are prepared in suspension in saline water. This is inoculated in the inoculum vessel located at the fermentation plant for the production of vegetative cells, because when we do the inoculation in the production fermenter, that should be vegetative cells, that should not be the spores, and these vegetative cells are used as the inoculum for the production fermenter. So here there is some important aspects of the morphology of the cells, that the success and yield of citric acid production mainly depends on the structure of the mycelia. The mycelia with fork and bulbous, that hyphae which aggregate the pellets and is ideal for citric acid formations. So this is that if the mycelia is loose and filamentous is limited branch, no citric acid production take place. So the mixing and shear stress is also crucial parameter. I mentioned before that one typical characteristics of citric acid fermentation or fungal fermentation process is that after at the initial stage of fermentation, you have to keep the agitator, mechanical agitator idle for some time. I can give the example that usually the inoculum vessel, the time of fermentation is 33 hours and your idle time is 10 hours, approximately 10 hours is the idle time. Now in case of production fermented, I told you that time is 124 hours to 144 hours. So and here the idle time is 30 hours. So this is the idle time. Here the idle time. This is the total fermentation time. So this time you have to give. If you give the time, then and only the mycelia will build up, and then the purpose of agitator to keep the cell in suspension in the fermentation media, so they freely interact with the media and give the products. So that is why it is required. So different morphology characteristic of the Aspergillus niger is this: biopellet at 5.5 after cultivation of 32 hours. This is, you can see, and free dispersed media at pH 3 after 24 hours is like this. More bunches is there, and morphology after the initial growth, pH as 3 for 8 hours, subsequent shifted to 5.5 and subsequent 24 hours is like this. So you can see how the morphology of the organism, they differ from time to times. Now if you look at the citric acid production process, it can be broadly divided in these steps. That the grouped raw materials we take suppose we take cane molasses here, then we do the upstream processing. Upstream processing means we had to include the air sterilization, medium sterilization, and pre-treatment of the raw materials or preparation of the raw materials. Then we take it to the fermentation process. Then after fermentation process, we get the group product, and through downstream processing we purify the product, we get the citric acid. Now this is what I want. This is the downstream processing of the purification process of the citric acid industry. The fermented liquor is we take it here. In the first I told you that in the previous slide I mentioned that I can show you there that here I told you that after the removal of that cell mass, we take it to the filtered reservoir, and after this we take this is the fermented liquor, this is the filtered reserve, filtered material, we take it here. This filtered material we treat with lime. Why we do treat with lime? Because when we treat with lime, lime is nothing but calcium hydroxide. Calcium hydroxide. This calcium hydroxide react with citric acid and precipitate in the form of calcium citrate, and this is the solid mass. So as soon as the calcium citrate is precipitated out, then we pass through the filtration. This is called panavis. I will show you how it works. Panavis, and we wash this with water, and then we discard the filtrate. Filtrate is the industrial effluent. And now I want to point out here that this is a typical type of effluent, because since we are treating with calcium hydroxide, this filtrate will contain lot of calcium ion. There is a possibility that filtrate, the effluent contains lot of free calcium ion. So this is the typical character, not only organic material but also calcium ion content will be very high. Then that the solid material calcium citrate, we take it to the treat with concentrated H2SO4, and then you react with H2SO4 and produce citric acid and gypsum, calcium sulfate 2H2O. and after that calcium sulfate is the insoluble mass. You pass through the Benavis the gypsum filter, where we can get the solid gypsum which is required for the cement industry. And then this filtrate contains the citric acid, with since it is produced from cane molasses, there is a possibility that it has lot of color, and then the citric acid crystal is perfectly clear. So it is required that we should remove the color from that. So we use the activated carbon just to remove the color, which has the bleaching property. And then this, after the treatment, we remove this calcium, we remove the activated carbon. I told you activated carbon is used very fine particles, and to remove the fine particles sometimes we use some kind of filter aid. Because one filter aid, I can tell you, for Solka flock, which is cellulosic material that absorb these fine particles on the surface. There is a bigger particle that we can easily separate it out. After that we concentrate it, we pass it through the evaporator, and it is the tubular evaporator. And because this content about 22% citric acid, this 22% citric acid, when passed through the evaporator, we get the 60% citric acid, and then we cool it down, and it undergo the crystallization take place, the solid crystal formation. Then we pass it through the centrifugation process. After centrifugation process, we separate out the crystals and mother liquor, that you take it back to the evaporator again just to concentrate further. And the solid crystal, you wash it first, and then we dry it with the help of drum dryer. There is a drying technique is there. Then we use some kind of sieving machine, so the different size of crystals can be separated out. So obviously the larger size of crystals will be having higher market value as compared to the small size of solid particles. Then we pack it in the polythene pouch and sell it in the market. This is the downstream processing of the citric acid industry. So I shall continue in the next class. Thank you.